like to say is that my work is greatly influenced by the smart people who are here at the table, the PhDs in science. Jane explained that she hosted an exhibition of mine quite some time ago in which I was documenting nature. I've been looking very closely at nature for a very long time. And at the time, it was looking at trees. And in some ways, my looking very closely at a cell or an egg, a spider egg, as you see in the film, is very much like the meticulousness that I would use in drawing. I'm now not just looking at trees as physical form, but now that I have equipment, the optical equipment available to me, be a scientist, I'm looking and documenting all of the orchids of Southeast Asia and translating their names from the Greek and Latin to English to understand what I was looking at. That those labels of the flowers explained to me that they were not the labels, they were not saying where the plants were from or anything about the plants, but it was the scientist's name. But I got very interested in museology, cataloging, and categorization, and began, of course, to look through the microscope as well. One of my deep fascinations with the microscope is that the invention of the optical lens by Antony van Leeuwenhoek in Holland, and then the microscope by Robert Hooke, was originally in order to do thread count and look very closely so that they could understand the quality of the cloth that they were selling. And it was by chance that they actually saw bacteria swimming in water. So I find this really interesting, and I have also engaged, as you can see in my exhibition, with working with material semiotics. That is, I like to use a lot of cloth and physical form. So what you're seeing in the exhibition is my desire to turn science into part of life. The chromosome painting piece is printed onto silk. The spider film is within a handmade paper enclosure. So that's often how I'm trying to translate between art and science. So I'm not sure I can completely answer Jane's question, but the desire for one to make the scientific discoveries readable and palpable to people around them, I think, is to put them out visually in a way that they can also experience them, because this is a secret world in a way that scientists are privy to. This is one of the first drawings of cell division, of chromosome splitting, meiosis, and mitosis, and I hand embroideries documented on them. This drawing happens to have been done by a female scientist. Also, I'm so happy to have you here, Amy, because there aren't a lot of women in the laboratories, and there certainly weren't a lot of women at Carnegie Mellon in the sculpture studio either, so very interesting. The next thing that happened in my life, from a personal standpoint, is I had a child with a genetic anomaly, and that really made me have to do a lot of genetic research and also changed my whole body of work. And after the subsequent loss of that child, my husband and I went through a lot of research. We did have a very healthy child, but the knowledge that we gained after the fact really has made me compelled to continue to understand what's the cutting edge of genetic research and why do we need to know it, or the cutting edge of science in general, so all sciences. But I was invited to the Wellcome Trust as a artist in residence in London, England at Gasworks, and this is the DNA sequencer that did the big job of sequencing the human genome. And the film that you're seeing in the space in the exhibition, I was allowed to use one of my colleagues' Astrofire microscopes in order to make that film. His name is Steve Black, and he studies gastralization and early development, and they're looking at spider eggs in order to understand human development and see the pacing of chromosome splitting and what goes wrong and what goes right, and when it does. He allowed me to make a 200-hour film with him and his assistant. And so what you're seeing is 10 films spliced together in the exhibition, and what I was trying to say with that, if I can cut to the chase with the exhibition, is that some of us live, some of us die, and it's only different pacings. And so what I was watching, as I was watching the development of these spider eggs, is that only a percentage of them made it through the whole series into becoming. And I wanted to have a film that actually documented that and also gives us the reality check of this is a factor in nature. It's not all just coming into being. And it's also quite stunning to watch that development as well. So making an installation, I'm interested in giving a kind of physical, architectural, phenomenological experience, so creating the paper walls like the skin on a cell, and I then edited and worked with them for actually a couple of years by the biophysicist Andrew Pelling, who was working at the time at UCLA's lab, and it's nanotechnology, and this is an AMF microscope that listens to the sounds of cells. And what you'll hear in the film is that cells that are sick sound quite differently than cells that are healthy. The cells that are sick sound like they're stretching, and I think our molecular biologists will be able to speak to this even more than I can, but the use of nanotechnology is amazing because it's a non-invasive way to detect sick or cancerous cells. So that's just a little bit about where my work's coming from and why I think it's, I guess, important to me to know what these people are doing. I use it maybe metaphorically, personally, but hope that it reaches people more globally than just my own story.